Excellent. So now we have the Translational Center for Barrier MPS. Jim McGrath will talk to us about breaking into ISTAND training and disease-agnostic tissue chip platform to speak FDA.
So thank you, Pasley, and thank you for this opportunity. We've done this a few times. These are amazing events, and we learned so much from them. So I'm here representing a team from both the University of Rochester, Duke University, and also RIT. Jonah Adamo, Hani Awad, and Ben Miller are co-PIs with myself on the University of Rochester team. And George Trusky is our lead PI at Duke University. And we also are partnering with RIT, and they've been a really important venue for us to do reproducibility studies and also advance some of the technical aspects of our chips. So I think one thing that distinguishes us from our colleagues in the TraCe cohorts is that we are really coming at this from a platform-centric perspective. So we're taking that platform, and it's been the mission for a while to apply that in as many contexts as possible. So we have at the University of Rochester developed a platform that involves silicon technologies for both membranes and also silicon sensors, which really give us unique capabilities. And we've put this together in a modular construction kit that allows us to change the design in order to fit specific tissue chip purposes. And we've got a core set of models that we've been working from and that are emerging now into our regulatory efforts. George Trusky's group is focused on muscle immune interface chips. Ben Miller and myself are working on neuroinflammatory injury models. Hani Awad leads synovial barrier remodeling chips. We have a number of chips that do immune cell trafficking metrics, and with the sponsorship through BNL, we're now working on blood-retinal barrier models as well. We've taken the opportunity to create a center at the University of Rochester for MPS very seriously, and really have taken a lot of what we're learning and doing in this context and really created a services arm for TraCe-bMPS. We do a lot of resource sharing. We have 21 labs that are participating using chips for various model building, 35 more projects or more, many chips going out every single year. And Jonah Damo's team has done a brilliant job in teaching us the quality system development aspects that we really need for the FDA engagement. We have more than 50 SOPs now that we've developed for various aspects and processes that are done on this chip, and we've worked through a reproducibility dashboard using our colleagues at Duke and RIT to really check our work in basic metrics on the chips. And we have a very large and growing network of partners, which I won't get into, but we're very grateful for. So we've been knocking on the ISTAND door now since the beginning, and we have not yet to get in. We've submitted 3 different LOIs, and the first 2 that we've submitted, one in 2024 and one in 2025, both came back non-reviewable. The first one is, I think, quite understandable. Now, I will say if you get turned away at this stage, the feedback you get from the FDA is not very helpful. It's a very cryptic couple of lines that you were then left with your colleagues to sit around and try to interpret and read between the lines, because we are interested in going forward. The first one I think is understandable. We put together an LOI focused on bone infection. This is a chip that had some predictive features. We were pretty proud of it, but it was for a class of drugs that were not yet developed. It was good at discovering mechanisms, but this is not something the FDA was at all aware of, and it was squarely an efficacy tool. Actually, initially going into this, all of our chips were squarely focused on efficacy. So we translated this brief feedback from the FDA and really sort of repositioned everything we do going forward. Now, in 2025, last year, we submitted juvenile dermatomyositis chips. This is focused on a rare disease. And here we actually still stuck with the efficacy positioning. And the reason for that is because we've heard FDA reviewers say that in clinical trials for rare disease, it's difficult to power these studies. And so human chips might actually be able to complement those studies. So interestingly, we also got this one bounced, but the reason it was bounced was because the drug class that we were selecting is not the primary drug class that's being used to treat juvenile dermatomyositis. That drug class is an immunomodulatory drug, and that was not the approach that we had taken. It was not rejected because of efficacy. So we're actively testing this hypothesis that the ISTAND program will accept an efficacy check in the context of a rare disease. So we're going back in after doing a few experiments and responding to this. The new one, which is the one I'm going to emphasize here today, tries to integrate everything we've ever learned and really take lessons from our colleagues, where you could see in the first two talks that preceded me, regulatory anchoring is part of the identification of the LOI that should go forward. So we just submitted this one. We have yet to be bounced. So this is also a little bit of a lobbying talk. So regulatory anchors. So it begins, I think, with the FDA roadmap, which says as a priority, they're going to phase out first monoclonal antibody therapies. So in fact, the monoclonal antibody, I missed a slide, I did. I missed the T-cell engaging slide. So first, the drug class. The drug class is T-cell engaging immunotherapies. And these are monoclonal antibodies that are coupling most successfully in hematologic cancers, coupling T cells to tumor cells in the blood. There's more than 100 of these in clinical trials now. There are 7 FDA-approved T-cell engaging monoclonal antibodies for blood cancers. And every single one of those comes with a black box warning with 2 parts: cytokine release syndrome and ICANS. So ICANS stands for immune effector cell-associated neurotoxicity syndrome. Every patient, most every patient that is treated with one of these therapies will experience CRS to some degree, and a good number of them will experience ICANS. And so this is just understood and managed as best it can be, but it's really not well predicted by the preclinical models, which I'll talk a little bit more about. So CRS is downstream, or ICANS is downstream of CRS. It's basically a septic storm of sorts, right? A cytokine storm that ends up in the brain and causing neurological damage. So regulatory anchors. So these are monoclonal antibodies, and again, their FDA roadmap calls for replacing these first in the transition to animals. So these are monoclonal antibodies, and in fact, the case study that is cited in the FDA documents for this priority is a T-cell engaging monoclonal antibody that nearly killed 6 healthy individuals in a first-in-human study. In both of those, this is the case study report that came out with it. You can see all these individuals that went into this trial experienced both CRS and ICANS. There's a lot of other FDA guidance on immunotherapy. Some of it's coming from CAR-T, which has the same 2 black box warnings. So we're able, and the details of these guidance documents actually recommend more complex preclinical models, including in vitro models with multiple cell types that are not used today. So we feel like we're really anchoring this in a place that the FDA is telling us they care about. So here's the context of use. The microsim CRS ICANS is a drug development tool to be used in the evaluation of safety of T-cell engaging monoclonal antibodies as immunotherapies. The tool will provide donor-specific risk assessments for both cytokine release and downstream ICANS, and will inform dose justification for investigational new drug applications prior to first-in-human clinical trials. And there really is a donor-specific aspect to this. You'll see we're using whole blood to generate the CRS in the chip, and so it's naturally a donor-specific response. So just quickly stop and just look at the lessons learned and how they show up already. Let me just make sure I'm blazing through this and I'm skipping slides. But we picked a drug class that the FDA is clearly watching. We framed a safety question, not an efficacy question. We aligned with regulatory priorities, and I'm going to try to convince you now we're filling a gap that no existing tool actually can fill. So here's the drug development use. The current paradigm is to do whole blood potency assays to find a dose that works, and then to go into the CRS. Whole blood is less common, but usually it's a spun down peripheral blood mononuclear cell cocktail that's treated, generates super physiological CRS. So this is not what the patients are going to see, but you can take the drug panels and controls and rank against it, and that's what's reported. The in vivo models are typically primates, which are famously not good at predicting the magnitude of CRS, and actually do not develop ICANS. So there actually is no ICANS prediction preclinically. So we're proposing to do the same potency, get the drug effective range in off the chip, but then to go onto the chip and take it through a process where we actually do dose evaluation and evaluate safety, because the CRS levels will come out in a range that is considered clinically safe, and also doesn't impact the downstream chip, which is the brain chip. So we will be able to provide a NOAEL for this class of drugs for the first time. So this quickly, I'm just going to take you through the technical description of how we're putting this together. There's a lot we can do. There's a lot we have done with elements of this. We really want to have the discussion with the FDA to make sure we're not putting a lot of effort into something that isn't desired. So that's one of the big lessons learned is we need to have pull on what we're doing. So we're going to generate the CRS upstream. It's a tandem chip. Use whole blood. Add cells that are not normally in the current assays, which will naturally be here. We'll add endothelium. They're known to contribute to the CRS profile. Neutrophils will be there, known to contribute to the CRS profile. The CRS profile will be filtered off and actually brought to the brain injury chip, and we've done a lot of work on brain injury metrics used for basically in the context of sepsis. So this really is in our wheelhouse. There's a lot of metrics we have available, but we're not telling you which ones we're going to use yet. We really want to have this conversation with the FDA to make sure we're building this in a way that makes sense to them. Here's some of the supporting data that we cite in the application. Brain injury, dose-dependent brain injury, activation, immune cell response, and is something we see in the chips, and then their ability to actually get these cytokine profiles inside the chips. This is courtesy of Ben Miller and his photonic sensors that are really being embedded into the same chips that we use. And we're also leaning heavily on well-established literature for this LOI in the context of CRS generation with the bispecific antibodies. So here's what the chip or the platform looks like. It's two chips. It's a CRS chip that's actually building up a profile, being filtered off as plasma, and then delivered to the secondary chip when it sort of peaks out. And so some of this is we've been able to lean on BARDA development efforts to create some of this, but the actual application is different, and so that we're able to leverage some of that to do to this. I also want to talk just quickly about the references and controls. We heard a little bit about that from the prior two speakers. There's FDA approved drugs which all produce some level of these toxicities. And so we're able to go through them, and we're able to source them and create a panel of low, medium, and high toxicity. And really our proposal is to be able to take any drug that comes in and sort of rank it here against this panel. We also have, because there's been drugs that have gotten into clinical trials and then pulled because of how dangerous they are, we also have some pretty powerful positive controls that we've got specified in the LOI. So that's where we're at. We've submitted a fresh letter of intent on this one. We are working on others and resubmitting the JDM letter of intent. We are working on the development. We are really moving ahead on this in project regardless, and it's going well. We want to thank CPATH. Not only are they organizing conferences like this, but they're actually partners in helping us sort of move through this. And actually, we credit to Pfizer, and thank you to Pfizer for being interested enough in what we're doing to meet with us quarterly and take a look at what we're doing and sort of guide it. And they are interested, which I think also is something I'd like to say to the FDA regulators in the room. They need a drug like this, they think. So thank you very much.